Good morning, everyone. Today we are going to continue with Chapter 2, the first document, Hereditary Traits and Genes. We've said that DNA is found in the chromosomes and chromosomes are found in the nucleus, and a specific sequence of DNA is transcribed into mRNA and the mRNA into protein, and we've said these processes occur by transcription and translation. Now we have genes. Every gene is responsible for a trait in the human being. And what determines a gene is the allele. The allele is a form of the gene. Today we are going to discuss this topic and take some examples. Document A, localization of genetic information. In this experiment, we are going to determine the location of genetic information. A true breeding black female mouse crossed with a male of the same genotype. It means a pure black female mouse crossed with a male having also a black genotype. The embryo formed of 16 cells was taken, and there was extraction of the nucleus of one embryonic cell. It means I take the nucleus of one of these embryonic cells. From the other hand, a true breeding gray female mouse crossed with a male of the same genotype. It means also a pure gray female mouse crossed with a male having also gray genotype. It means GG. The fertilized ovum was taken, having the two pronuclei, the male and female, and there was injection of the nucleus taken from this embryonic cell, from the embryo having 16 cells. Okay, now we have the nucleus of this embryo and the two pronuclei. These two pronuclei were extracted, it means they were removed. So now I have this ovum having only the nucleus of the embryo taken from the black mouse. Now 4 days in culture, in vitro, it means in the lab, the embryo has multiplication in the cells by mitosis. Then we have implantation of the embryo in the uterine cavity of a mouse which was not fertilized and it was ready for gestation, it means for pregnancy, but it is not fertilized. It is a white female mouse which is pure or true breeding. So now I put this embryo in this white mouse in order to develop and in order to have delivery or take birth from this white mouse. So now I have the birth of a black baby mouse. It means the result is a black mouse, not a white and not a gray. The conclusion from this experiment is that the nucleus carries the genetic information. It means not the fertilized ovum and not the uterus. It is the nucleus that carries the genetic information. It means I take the nucleus from the black mouse crossed together from the embryo of this mouse. Thus I have black baby mouse. So the nucleus carries the genetic information. Now we are going to talk about phenotypes and genotypes. A phenotype is the expression of genotype, and a genotype is the set of genes or alleles carried by the organism. Here I have a diploid cell. It means a cell having 46 chromosomes or 2n. I have here the gene which carries the alleles G or W. G is dominant, as you can see it is symbolized by capital letter, and W is recessive, symbolized by small letter. And remember, the dominant allele can be present as two copies like here GG or as one copy like here G, and it is represented by GW. The recessive can be only represented as WW, it means as two copies. Okay. And the dominant or the allele which is represented by capital letter dominates the second one. It means the second or the recessive is masked by the dominant. It means in the phenotype I have the expression of the dominant one. Okay, with respect to the genotype, I have first homozygote. Homozygote means pure, as we've said before, or true breeding. GG, I have the same alleles. Remember that homologous chromosomes carry the same genes but not necessarily the same alleles. For example, here they are same, but here they are not the same. But this is for the same gene. It means the same loci or same locus. Okay, here the genotype is GG, homozygote, while here it is heterozygote. It means different. GW, I write GW. The alleles are not the same. One is dominant and one is recessive. Here it is homozygote, WW. Okay, I write WW. This is the genotype. Okay, for example here, to compare phenotype and genotype, I write gray or G between parentheses for phenotype, while genotype is written as GG. You can write GG beside each other in the genotype. Okay, another one, it is gray, also having phenotype gray, but genotype GW. It is heterozygote, but also the phenotype is gray. It means those have the same phenotype but different genotype. Okay, white or W, I write in the phenotype. This is a white mouse. It is represented as WW. Now we are going to talk about true breeding lines and hybrids. In true breeding lines or selection, we have two mice having the same phenotype and the same genotype. Crossed together, they have black color coat. They are crossed together, they gave in the first generation all mice with black color. And in the second generation, also the mice are all with black color. It means in every generation, the phenotype and the genotype are the same as the parents. In the selection in the third one, also we have white mice having the same genotype and the same phenotype crossed together. They gave in F1 generation all mice that are white, and in the second generation all mice that are also white. It means in the selection we have the mice that are crossed together gave in F1 and F2 generation the same genotype and the same phenotype. This is true breeding lines or selection. In true breeding lines, I have the offsprings have always the same phenotype and genotype as their parents. While hybridization, it means I take mice with different phenotypes and different genotypes, but they are pure. It means here one is black and one is white. They are crossed together having BB and WW genotypes. It means they are pure. They gave in F1 generation all mice which have black coat color. It means black is the dominant. I conclude from F1 that black is dominant. And they are all hybrids. It means they have BW genotype. Okay, so in F1 generation they have BW or they are hybrids. They are all black. But in F2 generation I have different result. They are black and white. 3/4 black and 1/4 white mice. Thus the results differ in F2. So in hybridization the results differ. They are not the same as their parents because I have different phenotypes. Thus the results will be different. We will have white and black mice. This is hybridization cross. So a monohybrid cross is a cross in which we study the transmission of one trait or one gene. Okay, here for example we've taken T representing the allele for pea plant with purple flower and T small letter for the white flower. Cross between purple and white is done. TT capital letter cross TT small letter. The gametes here it gives T capital 100% and here it gives T small letter 100%. Of course, this is done by meiosis, the production of gametes. Random fertilization or by Punnett square, I have all TT, it means hybrids having purple color because purple is dominant. Okay, T from one parent and T from the other parent. So in F1 generation, I have all with TT genotype or purple phenotype 100%, because this is, as we've said before, a hybridization. It means two parents with pure genotype or true breeding but different phenotype, one is purple and one white. In F1 generation, all are hybrids with the dominant phenotype. It means T capital, they are purple.